It's Mrs. Esser, and today our topic is cell division, trying to answer the question of why do cells divide and what is the process to have division take place. So there's three main reasons cells divide. The first is reproduction. When I say reproduction, I do not want you to think sexual reproduction, not sperm cells, not egg cells, but instead, this type of reproduction would be considered asexual reproduction. So, this will be smaller organisms, such as unicellular organisms, bacteria, things of that nature, that will reproduce by binary fission or some form of asexual reproduction where they're making exact identical copies of the parent and then splitting into two. Another reason we need cells to divide is for growth. So hopefully you grew from a baby to a toddler to a little kid, preteen, teenager, and you will continue to grow throughout your life. But in order to do that, you need to make more cells. Increasing in size requires that. And then the last one is repair. So if you've ever broken a bone or fallen, and maybe you had a scab on your leg, repair is also one of the functions of cell division. You need new cells to grow and to take the place of those that were lost or broken. So how do our cells know when to divide? Our friend DNA always has instructions for everything in your body. So the DNA has instruction for the cell's activities of when to divide, how quickly to divide, and all of that. So with our cell division, it is important to note that DNA replication has to occur before the cell divides. So if you aren't confident in DNA replication, I'd take a look at that video. But every daughter cell has to have a genetic identical copy of the parent so that the cells are identical once they are fully divided. So taking a look at our pie graph of our cell cycle, just looking, we can see interphase is the cycle, the part of the cycle that it is in the longest. So this takes over 75% of our cell cycle. So this is going to be our preparation phase. So I'm going to talk about this on the next slide, but we have G1, S, and G2, where the cell is preparing for division and making sure it's ready. Our next part is mitosis, where the cell is physically dividing. So the four parts of mitosis are prophase, metaphase, anaphase, and telophase. And then the last part of the cell cycle is cytokinesis, where the cell is fully dividing into two. So just looking at our timeline, how long does it take? A human adult cell takes about 24 hours depending on the cell. We can see 18 to 20 of those are in interphase, so very long time preparing the cell for division, and then 2 hours actually splitting. You'll notice embryonic cells, so baby cells, things like that, before birth, I should say, are only 30 minutes. So we're going to talk more about embryonic stem cells in our biotechnology unit, but scientists love to study these cells because they replicate so quickly. So getting into our cell cycle, our first phase is interphase. So what happens here is our cell is going to double in size, so it's going to physically grow. Our DNA will replicate. As I said, that DNA replication video would be great to watch. Our organelles double in numbers. So right here, I don't have all of the organelles pictured, but the mitochondria, the ER, Golgi bodies, they're going to have to double in number. So here I can see my DNA in this form is called chromatin. It's loose spaghetti string-like DNA, so I can see that it replicated. I can see that my organelles replicated as well. So this is where I talked about our different parts of interphase, so G1, S, and G2. So G1 is the portion of interphase where our cell is increasing in size, making new proteins, making new organelles. Once our organelles have doubled, we go to S phase, which stands for synthesis. Remembering Stirringer's synthesis means to build or to create. So this is actually going to be DNA synthesis. We're copying, we're replicating the DNA, so we're going to have exact genetic information in the new cell. And then G2 is pretty much just the final preparation, making sure the organelles, molecules, everything we need are there for division. You'll notice on this picture also I have a little G0 here. So G0 could be a second loop where it's a temporary resting, or it could actually be a permanent spot where the cell just is no longer going to divide. So G0, think about going 0 miles per hour. You're going to pause the cell cycle, that's why sometimes the cell cycle takes a little bit longer. Next, going into mitosis or M phase. The M phase is made up of 3 parts: prophase, metaphase, anaphase, and telophase. So this is where our cell is physically dividing. So prophase, we're going to see our DNA form into chromosomes. So we had our DNA in that chromatin, that string-like spaghetti form. So that chromatin tightly wraps around histones to form a chromosome, so it's more tightly coiled up. Spindle fibers form, which I don't have pictured here, but they're going to be extending out of the centrioles, which are going to be responsible for eventually moving our sister chromatids to opposite sides. And then our centrioles will physically move to opposite poles and our membrane disappears. So this next click will be the picture of prophase. So I can see my membrane disappearing, we can see my chromosomes formed, and then the moving. So metaphase, I remember metaphase, my genetic material meets in the middle, meta meaning middle, so it's literally the middle phase. So our nucleus is totally and completely gone. This is the only time you'll see DNA outside of the nucleus. And the reasoning for that is because we need the DNA to be able to separate in order to have 2 separate daughter cells. And our chromosomes are going to line up in the middle or on the equator, middle. Then we have anaphase, where we're going to see our spindle fibers reach out and grab onto our chromosomes. So our chromosomes have this little part in the middle called a centromere, which holds them together. So those centromeres are going to separate because we eventually want these chromosomes to separate to each side of this cell. So chromatids separate, move towards opposite poles, so we can see the difference. Anaphase, they move towards opposite poles. That picture should have moved a little bit more. Telophase, nuclear membrane forms around each group of chromosomes. Our chromosomes unwind and cytokinesis can begin. So with telophase and cytokinesis, it's performed a little bit differently in plant and animal cells. So this here is an animal cell because I don't see a cell wall. I just see this outer cell membrane. So right here we call this a cleavage furrow. We can see where it's physically pinching in and cleaving the 2 cells. So telophase, I can see my nuclear membrane reforming, so my DNA is going to be stuck back inside of the nucleus, my organelles on opposite sides, so we're about ready to fully split the cell. So that's where cytokinesis takes place. The cytoplasm fully divides. 1 cell becomes 2 identical daughter cells. So as I said with cytokinesis and telophase, it's a little bit different in plant versus animal cells. So animal cell, as I said, that cleavage furrow, it's pinching in half. But we know plant cells don't do that because they have a very rigid outer cell wall made out of our polysaccharide cellulose for structure and support. So instead, you'll see in the middle the cell plate forming, and that cell plate will eventually become a new cell wall. So that is our process of the cell cycle. A little trick to the tray that helps me always remember it: interphase, prophase, metaphase, anaphase, telophase, cytokinesis. I remember it. I practice math at the cafe. So that's just a little trick to help remember the order. Once you're done separating the 2 cells, cytokinesis, your cells might take a break or they might go right back into interphase and continue to dividing. So it's a constant cycle of the cells dividing. So that's it for our cell cycle.